Hello everyone, this is your educator Priyanka Thakur. I teach you life sciences and welcome you all on our YouTube channel GurMantra Shikshaka Granth. Today you're having a practice session on nucleic acid, supercoiling and replication. So these are the topics we have been covered in a few days. You might have seen the video of transcription, but I'll be taking a practice session on transcription translation separately during some other day. I hope you all have you all have had a great Diwali, but if somehow due to some reason you fail to enjoy this Diwali to that extent as much you were expecting, so do not worry, just work hard, achieve whatever you want to achieve in your life so that you can celebrate every day as a Diwali. Okay, I hope you understand my intentions. So let's start with our practice session. The total marks are 30. Each question will be carrying 2 marks. A whooping negative marking is there, it's 50%. So please go and bring your pen and copy. Please bring your pen and copy. Okay, not for me but for yourselves. So the time given is 45 seconds. Okay, I haven't placed any timer in the video, but I'll be telling you the time. So it's 45 seconds. So you have to be honest with your answers. Now it depends upon you because I'm not seeing you. Okay, so it all depends upon you. Either you want to be honest or not, but just be honest. Definitely do not just forget to comment your score. Tell me your score in the comment box. Please tell me your score in the comment box. Okay, so all the very best to all of you, and here is your first question on your screens. So just wait for 2 seconds. You will be having your first question on your screen soon. This is your first question and your time start now. If you need more time, you can pause the video, solve the question and then comment down your answer. Write down your answer. Fifteen seconds. 4, 3, 2, 1. Time's up. Okay, so here's the question. A bDNA, which is double-stranded bDNA, it has 1500 base pairs. Out of these 1500 base pairs, 70% of purines are adenine bases. They are asking you the total number of hydrogen bonds in the DNA. Now remember, 1500 base pairs means 3000 nucleotide. And out of those 3000 nucleotides, the purine adenine is not out of those, the total amount of purine. Out of that total amount of purine, 70% will be of adenine and 30% will be of guanine. It means if adenine is 70% and then thymine will also be 70%. If guanine is 30%, then cytosine will also be 30%. So we understand that out of total number of nucleotides, 70% are of adenine and thymine. So just do it 70% of 3000. You will get your answer. How much? You will get 2100. It means 2100 nucleotides are of adenine and thymine. 1050 will be of adenine. 1050 will be of thymine. And remaining 900, 450 will be of guanine. 450 will be of cytosine. So 1050 adenine will be bonded to 1050 thymine by 2 hydrogen bond. So total number of hydrogen bond present in between adenine and thymine are 2100. Okay. So 450 guanine will be bonded to 450 cytosine with the help of 3 hydrogen bond. So there might be about 1300 50 hydrogen bonds present in between 450 guanine and cytosine base pairs. The total number of hydrogen bond become 2100 plus 1350, and which comes out to be 3450. So here is your answer, D. I hope you get my answer and you had your pen and copy and you were writing it down. Okay. So the next question on your screen is here and your time starts now. Twenty seconds. Five, four, three, two, one. Time's up. DNA application is semi-conservative, 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 semi-conservative. Fine. DNA application is continuous, bidirectional, discontinuous, unidirectional. Now see, DNA application is not continuous. Most of the time it is not continuous, it is discontinuous. But it can be bidirectional or unidirectional. So the appropriate answer is DNA application is semi-conservative and discontinuous. So your answer is C. So here you have your next question on your screens. Your time starts now. I'm giving you 45 seconds. Twenty seconds. Five, four, three, two, one. Time's up. Okay. Which of the following subunits of DNA polymerase 3 act as clamp loader? Alpha complex will be forming the core enzyme alpha theta epsilon. Delta, there is no such complex. Delta comes under the gamma complex. Tau dimer, it will be attached to the core enzyme. So here the answer is gamma complex. So your answer becomes D. The first question I have discussed earlier, it has been asked in GATE earlier. So here is your next question on your screens and your time starts now. Nineteen seconds. Five, four, three, two, one. Timed up. Okay. Which of the following reaction is required for proofreading during DNA application by DNA polymerase 3? 5' to 3' exonuclease. See, the students who are attending my classes regularly will be able to answer all the questions without doing a single mistake. Okay, so DNA polymerase 3, 5' to 3' exonuclease activity, this one is wrong, because DNA polymerase 3 does not possess 5' to 3' exonuclease activity. Next is 3' to 5' exonuclease activity. So this one is right, and DNA polymerase require 3' to 5' exonuclease activity for proofreading. and endonucleases activity is not there. So the answer of your fourth question is B. So next question on your screen, and your time starts now. Fifteen seconds. Okay, you are saying that we are done. Do not worry, you still have to wait. 3, 2, 1, time's up. Which of the following enzymes remove supercoiling in replicating DNA ahead of replication fork? Is it DNA polymerases, helicases, primases? DNA polymerase will not remove supercoiling. It will be responsible for the polymerization of DNA. Helicase is responsible for breaking your hydrogen bond, but still supercoiling is removed by the breakdown of phosphodiester bond. Primases will be adding a primer, so the answer is topoisomerases, which relieve the supercoiling ahead of the replication fork. So your answer is D. Next question on your screens. Time starts now. Fifteen seconds. 5, 4, 3, 2, 1, and time's up. What is the reaction in DNA replication catalyzed by DNA ligase? Addition of new nucleotide to the leading strand. Again, second option is addition of new nucleotide to the leading strand. Both of these options are wrong, same and wrong. Formation of phosphodiester bond between the 3' hydroxyl. Even if there is addition of new nucleotide to the lagging strand, it will be wrong. Okay. Formation of a phosphodiester bond between 3' hydroxyl of one Okazaki fragment and 5' phosphate of next on the lagging strand. Okay, this is the function of DNA ligase, base pairing of template and the newly formed DNA strand. No, it will not base pair in between template and newly formed DNA strand, which is not even required. So C is the correct answer, because DNA ligase is responsible for the formation of phosphodiester bond, and it will be forming the phosphodiester bond between the 3' hydroxyl of one Okazaki fragment and 5' phosphate of the next nucleotide. So the answer to this question is C. And here you are having your next question on your screen, and your time starts now. Twenty seconds. 5, 4, 3, 2, 1. Time's up. Okay. So, which of the following statement is true? Sugar component of nucleotide is ribose. This one is right. Sugar component of a nucleotide is deoxyribose. This one is also right. The bases in nucleotides are attached to a pentose sugar moiety by a glycosidic linkage. Again right. The sugar molecule of the nucleotide is an L configuration. This one is wrong. So you might be thinking, ma'am, you should have asked which of the following statement is wrong. It's not like that, dear students. The sugar molecule of nucleotide is in D-dextrorotatory configuration, but the sugar component in nucleotide may be ribose or deoxyribose. The DNA and RNA have not been mentioned here. So our correct answer becomes C, in which which states a nucleotide are attached to pentose sugar moiety by a glycosidic linkage. So your answer is C. Next question on your screen, and your time starts now. Ten seconds. And time's up. Which of the following is true about phosphodiester linkage? 5' phosphate group of one nucleotide unit is joined to the 3' hydroxyl group of next nucleotide. Is this the correct one? Or 3' phosphate group of one nucleotide unit is joined to the 5' hydroxyl group of the next nucleotide. See, we are sure about that 5' phosphate will not be joined to the 5' hydroxyl group, and 3' phosphate will also not join with the 3' hydroxyl group of the next nucleotide. So answer could be A or B, but we are sure about this, that a 3' group does not of a nucleotide does not have any phosphate group. So answer is A. And here is your next question on your screen, and your time starts now. Ten seconds. Time's up. When there is no net binding of DNA excess upon itself, the DNA is said to be in elongated state. No, it's not in elongated state. Compressed state. No. Relaxed state is the right answer. If there is no net bending of DNA excess upon itself, it is said to be in relaxed state. Okay. So answer is C. Okay, you can see the answer is always C. So you can blindly guess that answer is going to be C for the next question as well. So here's your next question on your screens, and your time start now. Ten seconds left. So time's over. During hyperchromic shift, the relative absorbance of DNA, it actually increases by 40%. Hyperchromic shift means increase in the relative absorbance of DNA at 260 nanometer, and the absorbance increases by approximately 40%. So answer is again C. Okay, so here you are having your next question on your screens, and time starts now. 5, 4, 3, 2, 1. I know you already had answered this question. Before that, I say time's up. So what is the main function of tRNA? I do not want to discuss proofreading or inhibit of protein synthesis. So here's the direct answer. Again, C. So C is your answer. Next question on your screen. Time starts now. Ten seconds. 2, 1, times up. Okay. How does Ethidium bromide interact with DNA? Does it bind with adenine? No, it doesn't. It binds with phosphate? No, it doesn't. It intercalates between the bases. Yes, it intercalates between the bases and decrease the stacking in between the bases, and it can cause the negative supercoiling. It can relax the positively supercoiled DNA. So C is again the right answer. Okay, so next question on your screen, and your time starts now. Ten seconds. 4, 3, 2, 1, times up. See, N15 radioactive ammonium chloride is switched to a medium containing N14 ammonium chloride for 3 generations. What will be the ratio of hybrid N15 N14 DNA to light DNA N14 at this point? See, here what you have to do, N15 DNA. You got to know that it is N15 DNA. Okay, so it has been switched to a medium having a N14. Okay, so it will be grown there for 3 generations. So make the 3 generations of DNA according to semi-conservative model of replication, and then find out how many hybrid DNA are there, how many light DNA are there. Do the ratio and you will get your answer A. So if you have had answered C, it's gonna be wrong. Here the answer is A. If you couldn't attempt this question, if you fail to do it rightly, so you can comment down in the comment section. I will tell you how to do it again. Okay, so fine. Here is your next question on your screens, and your time starts now. 3, 2, 1, times up. What is the role of FEN? Does it break down the glycosidic bond or hydrogen bond, or remove the first nucleotide of RNA primer, or it break down the phosphodiester bond? So you should know that in a case of prokaryotes, it's DNA polymerase 1 which will remove the primer. So you think the answer might be C now. No, it's not like that. In case of eukaryotes, RNase H removes the primer, but it failed to break the bond between the last nucleotide of primer and the first deoxyribonucleotide. So there must be a phosphodiester bond. So answer become D, not the first nucleotide. It will remove the primer, but it will remove the last nucleotide of RNA primer and not the first one. So answer becomes D. So I hope you have given me a correct answer here. So last question on your screen and your time starts now. Twenty seconds. 10 seconds. 5, 4, 3, 2, 1. Time's up. Peptidyl transferase is found in 60S subunit of ribosome, 28S rRNA or 23S rRNA or all of the above. Remember, peptidyl transferase is found in the larger subunit of ribosome. 60S is also larger subunit. 28S rRNA is a component of larger subunit, and in 60S rRNA, 20S in 60S ribosome, 28S rRNA will be having this peptidyl transferase, and 23S rRNA is a component of 50S subunit of 70S ribosome. So all of these will be having this peptidyl transferase. So answer to this question is D. This is all for today. Hope you liked it. So please like the video, do subscribe to our channel, join us wherever you want, and keep sharing the video. Wait for the next sessions too. Thank you. Stay safe, stay healthy.